Hello everyone. Today we are going to discuss about life history or life cycle of Penicillium. Okay. So first of all we will going to discuss what is the systematic position of the Penicillium. Systematic position, it is included in the kingdom Fungi, subkingdom Dikarya, class Euromycete, order Eurotiaceae, family Eurotiaceae and genus Penicillium. Okay. This much you have to remember. Nothing to understood in this. This is the systematic position of the Penicillium. Then we are going to discuss about what is its nature and vegetative structure. In nature, some Penicillium are parasitic in nature. Some species are parasitic in nature, and some are also saprophytic in nature. Saprophytic in nature. Okay. Some species of Penicillium are parasitic and some species are saprophytic. What is the meaning of parasitic? Parasitic means those which grow on other organism and this Penicillium is only benefited and other one is harmed. And saprophytic means when the Penicillium is grow on dead and decaying materials. Okay, some species are parasitic and some are saprophytic in nature. So this is the nature of the Penicillium. Then it is generally called green mould. Okay, remember this one. Penicillium is also generally called green mould, mould, and it is best known for the discovery of first antibiotic penicillin. Okay, so this is also generally known as green mould. Okay, this is the nature of the Penicillium. Then we are going to discuss about what is the vegetative structure of Penicillium or Thallus structure, you may say. Vegetative structure or Thallus structure. Thallus structure of Penicillium. Penicillium is a mycelial fungi. That means the body of the Penicillium or the thallus body of the Penicillium is made up of mycelium. Okay. These mycelium are made up of hyphae. Okay. Suppose this is the hyphae. Now, the hyphae of Penicillium is septate. That means septa are present and these septa consist of a single pore. Pore is present due for the intermovement or inter-exchange of cellular protoplasm and cytoplasm, other materials, etc. etc. So the cell are septate. The cell wall is present. Cell wall is made up of chitin. Right in this way. So in thallus structure, the Penicillium is a mycelial fungi. The body of the Penicillium is made up of mycelium. The myceliums are made up of hyphae. These hyphae are septate in nature. Septa, in the center of the septa, a hole or pore, pore is present for the inter-exchange between inter-exchange of cellular materials. Okay. Then the cell wall is present and that cell wall is made up of chitin, etc. etc. So this is the thallus structure. It is the hyphae. Okay. These hyphae are septate like this. These are the hyphae. These are the septate hyphae. Okay. This is the general structure of the Penicillium. Next, this is the thallus structure. Okay. Then there is reproduction. After thallus structure, there are reproduction. This reproduction is very important. In reproduction, Penicillium reproduces by two methods. One is asexual method, another is sexual method. Asexual method and sexual method. Asexual method of reproduction occurs due to presence of conidia, conidia. Asexual reproduction occurs by means of conidia, spores called conidia, asexual spores called conidia, and sexual reproduction occurs by process plasmogamy, by process called plasmogamy. Okay, now we are going to discuss about first the asexual reproduction of Penicillium. It is very important, always frequently asked in question. Asexual reproduction. asexual reproduction by means of conidia. Okay. Asexual reproduction occurs by spores called conidia. These are the spores called conidia. The conidia spores are formed in vegetative branch called conidiophore. Asexual vegetative branch called conidiophore. PHOR. Conidiophore. Inside this conidiophore, conidiospores are formed. Conidiospores are formed, and these conidiospores are nothing but the conidia. Okay. So we are going to discuss about the structure of the conidiophore. See, this is a thallus. Thallus, or you may say hyphae. Hyphae is the plural form, hyphae is the singular form. This is the hyphae of the Penicillium. Now, the conidiophore arise from any vegetative hyphae. Okay, as this is septate, septa is present, septate hyphae. That means the hyphae are separated from each other. This is one hyphae, this is another hyphae. So, in the separated hyphae, the conidiophore arise from any hyphae. Suppose this is a hyphae. From this, a conidiophore is arise. These conidiophore are also separated by septa. They are uninucleate to multinucleate. Uninucleate to multinucleate structure. Okay. This is the conidiophore. This is the structure of conidiophore. Now what's happened? In the structure, the thallus is aerial thallus, and the part of the thallus is aerial, and some part of the thallus is penetrated inside the substratum. Okay. Penetrated inside the substratum or the host cell. A part of the hyphae is present below to the substratum or penetrated inside the substratum, and a part of the hyphae is aerial, that means present above to the substratum. From this aerial part, aerial substratum, the conidiophore arise, and this is conidiophore. Okay. Now, at the apex of the conidium, some bottle-like structures are bottle-like structures are present. Like this. Some bottle-like structures are present. These bottle-like structures are called sterigmata. Sterigmata. Bottle-like structure, BOTTLE. Bottle-like structures are present at the apex of the conidiophore. Those are called sterigmata. Okay. Now, this is the one hole of sterigmata. Hole. WHORL. This is the one hole of sterigmata. And this conidiophore is non-branched. It is unbranched. There are some conidiophores present which are branched also. See, this is the main branch, or it is called the primary branch of the conidiophore. From this primary branch, secondary branches are arise. See, this is the primary branch and this is the secondary branch. Okay, these secondary branches are called medulla. Okay, this is the secondary branch called medulla. METULA. Medulla is the singular form. Medullae is the plural form. If you add E, then that will be the plural form. Okay. The secondary branches of the conidiophore are called medullae. Plural form is medullae, singular form is medulla. Okay. Now, here it is one hole of sterigmata is present, but sometimes what's happened? Two or more than two hole of sterigmata are also present in one hole, in one conidiophore. Okay, see, this is two hole in two branches. Okay, then conidiophore, if branches occurs, then those branches are called medullae. If branches are not occur, then at the apex bottle-like structures are present. Those are called sterigmata. Then to this sterigmata, conidiophores are produced. These conidiophores are produced in basipetal manner. Conidiophores are produced. These are the conidia or conidiospores. These are the conidiospores. Okay. These conidiospores are also called conidia. C-O-N-I-D-I-A. Conidia. Okay. These are present or formed in basipetal manner. B-A-S-I. Basipetal. Basipetal manner means youngest are formed at the base. Okay. Youngest are at the base. Y-O-U-N-G-S. Youngest or newly formed are present at base. And older one at the apex. Older conidia are present at the apex, and newly formed or youngest conidia are present at the base. This is called the structure of the conidia. Now these conidia are structurally ovoid. Conidia are structurally ovoid, ovoid or globose. Okay. Now if you see, the structure of this conidiophore is look like a brush. B-R-U-S-H. This is the handle of the brush. This is the plastic of the brush. Okay. So if you see, this looks like the structure of a brush. That's why it is known as Penicillus. Brush is also known as scientifically Penicillus. That's why from this Penicillus the name of the fungi Penicillium is derived. Okay. So this is the structure of the conidiophore. In conidiophore, conidiospores or conidia are arise when these mature conidia are detached from the conidiophore and falls on the substratum. They start germinate. First, a germ tube is developed. This is the germ tube of the conidia, and when this germ tube penetrates in the substratum, that will form a new hyphae. From this conidia, new hyphae are formed. Okay. This is the method of the asexual reproduction. Asexual reproduction occurs by means of conidiophore. Conidiospores are arise in the conidiophore. Structurally, conidiophore arise from any aerial hyphae in the upward manner. Then this may be branched or unbranched. If branches are present, those branches are called medullae. Then at the apex of the conidiophore, some bottle-like structures are present. Those are called sterigmata. In these bottle-like structures, some spores are present, formed. Those spores are called conidiospores or conidia. When these conidia are detached from the conidiospore and fall on the substratum, then they grew into new hyphae. This is the asexual method of reproduction. Then we are going to discuss sexual method of reproduction. Sexual method of reproduction. Sexual method of reproduction is occurs by a process called plasmogamy. Plasmogamy. Now, the literal meaning of plasmogamy is fusion of protoplast. Okay, literal meaning of plasmogamy is fusion of protoplast. How this fusion of protoplast help the sexual method we are going to discuss. Now, the male sex organ and female sex organ. The male sex organ of Penicillium is called antheridium, and female sex organ of Penicillium is called ascogonium. Okay, this is a singular form. Antheridium, ascogonium singular. Antheridia, ascogonia are plural. Okay. Now these sex organs are arise from any vegetative part or any part of the hyphae, any aerial part of the hyphae. How? First, ascogonium is developed. First, ascogonium is formed in upward manner from any part of the hyphae. This ascogonium we are going discussing first the ascogonium. Okay, ascogonium arise from any vegetative hyphae. They are generally formed uninucleate, but they grew into divide and redivide to form 32 to 64 nuclei. Okay, when they newly formed, they are formed as uninucleate condition. Single nucleus is present, but after their growth and development, that single nucleus is divided to form 32 to 64 nuclei. Okay, then near to the ascogonium in the same hyphae or in other hyphae near to the ascogonium, Antheridium is formed. Antheridium is formed. Okay. Now this Antheridium is also septate and multicellular. This ascogonium is multicellular, multinucleate. This Antheridium is also multicellular and multinucleate. This Antheridium, after development, coils around the ascogonium. Okay, like this. Coils around the ascogonium. Coils around the ascogonium. Now, after the coiling, the tip of the ascogonium, the tip part of the ascogonium, detached from the main body part, and large amount of protoplast and nuclei deposited at that tip of the Antheridium. Okay. After the Antheridium coils around the ascogonium, the tip of the Antheridium, in the tip of Antheridium, a large amount of protoplast and nuclei present, and that is separated from the main Antheridium part. Now what's happened? After its growth and development of Antheridium and ascogonium, the intercellular space, intercellular between Antheridium and ascogonium dissolves. The intercellular between Antheridium and ascogonium dissolves, dissolves, dissolves or break. Okay, if I do it in large form like this. Okay, this is the tip of Antheridium, this is ascogonium, this is the intercellular space, intercellular. This intercellular is dissolves. After the dissolvement of the cell wall, the two protoplasts of Antheridium and ascogonium intermixed. Okay, but in this intermixed, only the protoplast of Antheridium and ascogonium mixed. The nuclei of the Antheridium does not enter into the ascogonium. It is very important. Okay, that's why it is known as plasmogamy. Only the protoplast of Antheridium or protoplasm of the Antheridium mixed with the protoplasm of the ascogonium. Not the nuclei or any nucleus of the Antheridium enter or penetrate into the ascogonium. Okay. Now, this intermingling or fusion of protoplast between Antheridium and ascogonium create a stimuli which cause these nuclei of ascogonium, which cause the nucleus of the ascogonium present in diploid condition, which cause the nucleus of the ascogonium to be present or to be formed a diploid state. The single nucleus of the ascogonium fuses with each other to form a diploid condition, diploid nucleus. These diploid nucleus are now called dikaryon, and the process by which this dikaryon is formed is called autogamy. Okay, try to understand. After the fusion of the protoplasm or the protoplast, after the fusion of protoplast, the nucleus or nuclei of Antheridium does not enter into ascogonium. This fusion of a protoplasm cause the nuclei of the ascogonium to be present in a divalent or dikaryon form, in diploid form. They intermixed with each other. That intermixing nuclei is called dikaryon, and the process is called autogamy. Okay. Now what's happened? This dikaryon present inside the ascogonium. This is the dikaryon stage. These dikaryon formed ascogenous initial cell. Okay, these dikaryon form a single dikaryon form a single ascogenous initial cell. A single dikaryon form a single ascogenous initial cell. These ascogenous initial cell form ascogenous hyphae. Ascogenous hyphae, HYPHAE. Okay, these ascogenous initial cell, growth and development occurs to this ascogenous initial cell and they form ascogenous hyphae. Okay, these ascogenous hyphae then form asci, 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 or singular is called ascus. It is very important, always asked in question. Ascogenous hyphae form asci. Okay, now this asci or ascus, if you say asci then are, asci are the diploid stage of Penicillium or ascus is the diploid stage of Penicillium. Okay, this is the ascus, this is ascus. Mark here, this ascus or asci, asci are the diploid stage of Penicillium or ascus is the diploid stage of Penicillium. Okay, now these asci, the diploid nucleus of asci, the diploid nucleus of asci undergo one meiotic division followed by one mitotic division to form ascospore, ASC, ascospore, SPOR, ascospore. These ascospores are haploid in nature. Okay, see, this is, suppose this is one ascus having diploid nucleus. This is undergo first meiotic division. In meiotic division 4 nuclei are formed. This is undergo then first one mitotic division. In one mitotic division 4 nuclei formed, 8 nuclei, 1 2 3 4 5 6 7 8 nuclei. Now from the diploid asci, diploid from one diploid ascus, 8 haploid ascospores are produced. 8 haploid ascospores are produced. Okay, when these ascospores their growth and development complete, they detach from the main plant and fall on the substratum. Which one? Ascospores. These ascospores, when growth and development completed, they detach from the main plant and fall on the substratum and grew into new hyphae. Okay, this is the process of sexual reproduction. Sexual reproduction occurs by plasmogamy method. It is occurs by fusion of protoplasm only. The male sex organ is called antheridium and female sex organ is called ascogonium. Female sex organ develops first. It is initially a unicellular structure, uninuclear structure. But after the growth and development, it formed into 32 to 64 nuclei. Then nearer to the ascogonium, antheridium is developed and it coils around the ascogonium. Then the tip of the antheridium is separated by depositing a large amount of protoplast and a nuclei. Then the intercellular space or the intercell between ascogonium and antheridium dissolves. Then the fusion of a protoplast between antheridium and ascogonium occurs. But after the fusion of protoplasm, the nuclei or nucleus of antheridium does not penetrate into the ascogonium, but this create a stimuli in which the nuclei of the ascogonium arrange in a diploid condition or dikaryon. That condition is called dikaryon and the process is called autogamy. From these dikaryon, ascogenous initial cells are produced. From this ascogenous initial cell, ascogenous hyphae are produced. From this ascogenous hyphae, asci are produced. Asci are the diploid stage of the Penicillium. From this asci, the ascospores are produced by when the diploid nuclei of the asci undergo one meiotic and one mitotic division, they form ascospores, haploid ascospores. From one ascus, 8 haploid ascospores are produced. When these ascospores are detached from the main hyphae and fall on the substratum, then they grew into new hyphae or new penicillium hyphae. This is the sexual reproduction of the penicillium. Okay. Then their life cycle. Simply life cycle. Life cycle of penicillium is called haplophagic type. Phagic type. Remember this one. Okay. The life cycle of penicillium is called haplophagic type. Okay. This much. In this class, we will discuss about the life cycle of penicillium. What is the structure? What is their reproduction? Asexual and sexual reproduction of penicillium. Okay. Thank you everyone. Thanks.